I'd like to thank the CED team for the opportunity to showcase Symberix today. I'd like to introduce the company with a simple question. What makes us human? Based on Dr. Boyer's presentation, the answer might seem obvious. It is our DNA that makes us human. Well, there's an important clarification that needs to be made. The human genome contains only 1% of the total genes in the human body. The other 99% are not human. They're, in fact, the microbiome, the trillions of bacteria that live with us. The microbiome is important for human health and disease. Maybe someday it'll be considered part of our DNA because it is, in fact, a part of what makes us human. Symberix was founded based on a new idea, that the microbiome can be selectively targeted to improve human health. The original technology was licensed from the University of North Carolina based on innovative research conducted by the scientific founder. Our business case is built on 3 value propositions. We have the first valid microbiome drug target. Our drug discovery efforts are focusing on identifying 2 potential first-to-market products for 2 lead indications. And we believe that our technology allows for future expansion to other indications. So I'm a scientist by training, and I have to understand the science to believe it. So this is how our science works. We are targeting a bacterial enzyme responsible for the toxic side effects of some pain and cancer drugs. Specifically, the enzyme is located in the intestinal microbiome. The enzyme is called GUS, and there are just 2 things one needs to know about GUS to understand the science. One is it has a sweet tooth. It chews a sugar, just one kind of sugar, the glucuronide. Second is it is non-essential bacteria and not harmful to us, except when it comes into contact with certain drugs that the liver has detoxified by coating it with the very same sugar that this enzyme ultimately chews off. So you can imagine what happens. The drug is reactivated in the lower GI tract, and this causes severe intestinal damage. So 2 examples of drugs that exhibit this mechanism of toxicity is the cancer drug irinotecan and essentially all non-steroidal anti-inflammatory drugs. And their interaction with GUS produce a number of serious side effects. Our therapeutic approach is simple and elegant. To identify inhibitors that block GUS, block the toxicity without harming the microbiome. So this work was done over the last few years, and a number of publications have come out, including science papers showing using irinotecan as a proof of concept that this therapeutic idea works. So what we're proposing here really is a new paradigm of microbiome-targeted drug discovery. There are no drugs approved that work by selectively and safely targeting the microbiome. There are just 4 rules we follow in our drug discovery efforts. The target must be disease-relevant. Our molecules must be potent, selective, and not harmful to the microbiome. So we've identified 2 lead chemical scaffolds that fulfill all 4 conditions, and our current effort is in the lead optimization stage to identify at least 1 IND candidate. So before sharing with you our R&D timelines, I want to briefly walk you through the 2 lead indications here. 1, diarrhea is one of the most feared and intolerable side effects of chemotherapy. Patients require burdensome intervention treatment for their diarrhea, and a patient may suffer multiple episodes during a full course of chemotherapy. Our initial focus is irinotecan, which is given to treat advanced colorectal and pancreatic cancer. So this is an important chemotherapeutic agent with diarrhea as its dose-limiting toxicity, and therefore is the primary reason patients need to reduce their dose of chemotherapy. Our therapeutic goal here is to do 2 things: prevent and reduce the severity of the diarrhea, and allow patients to resume a full course of chemotherapy. Non-steroidal drugs, as you may know, are probably the most prescribed and used medications in the world, and we all are aware of the upper GI side effects of NSAIDs. But in the last 10 or 15 years, the medical field has come to realize that the lower GI side effects are probably as important. They result in asymptomatic toxicities such as enteropathies, And for chronic NSAID users, about 40,000 patients are hospitalized a year for NSAID toxicity. The FDA has identified this to be a major concern, partly because none of the drugs they've approved for the upper GI actually work for the lower GI. In fact, they may actually further harm the lower GI. So we have two, so the total market for these two products is about $1.5 billion. There are no FDA-approved products for either of them. And our goal is to develop two drug products as therapeutic adjuncts, one for irinotecan and the other for use with NSAIDs. So our drug discovery strategy is essentially file an IND in 2017 based on two product profiles shown here and a potential first exit opportunity with clinical data by the end of 2017. So we've received $3.5 million research prior to Symberix, and the company has received $300,000 in non-dilutive funding, including a phase 1 SBIR grant. So that concludes my presentation. We have a table in the next room over, so please come join us and learn a little bit more about what makes us human. Thank you.